Hello, students, and welcome to my video on cell structure and function. This will cover topics 2.1 through 2.3. We'll go on a tour of the cell, and we'll learn about why our cells small. Like all my videos, this is not for profit, cannot be used commercially. It's just meant to help students who find it on the internet. Here we go. I find it useful to look at the curriculum, look at what the College Board is telling us as teachers we need to teach you, what you need to know as students, and there's learning objectives and essential knowledge pieces, right? If you can really describe them, then you're doing well in the course. Plus, these are cool. They have some illustrative examples that your teacher might go over in class. So when I look at 2.1, it's telling me cell structure, the components of the cell. And my objective is to describe the structure and or function of these components and organelles. And 2.2 is going to do the same thing, but say, how does it contribute to the function of the cell? So let's look at the key terms here. I see ribosome. I see endoplasmic reticulum. I see rough and smooth endoplasmic reticulum. I see Golgi complex or the Golgi apparatus. I then see an exclusion statement that I don't need to know how the Golgi makes specific phospholipids, the packaging of specific enzymes for lysosomes or other ones. That's beyond the course. So that's good to know, especially if I have like a dense textbook I'm studying. The mitochondria, the lysosomes, the vacuole, the chloroplasts. Many of you have probably had these organelles in an introductory biology class, and some of you, this might be the first time. And so we're all coming at it from different angles, but we'll figure it out. One important thing to remember is that all these organelles are made up of carbohydrates, proteins, lipids, nucleic acids, the four major macromolecules that we looked at in Unit 1. In other words, they make these organelles. And just like we talked about emergent properties with water, how the whole is greater than the sum of the parts, the same here. All these organelles together make the property of life for the cell. The cell is able to divide. The cell is able to process energy. The cell is able to reproduce. The cell is able to maintain homeostasis. All those characteristics of life due to the emergent interactions of these organelles. We can't, at least in my class, I'm going to say we can't really skip over bacteria, right? So it started with all these subcellular components, but we evolved from bacteria. And so here is a prokaryotic or a bacterial cell. And we can see that the leuca was bacteria, right? And then eukaryotes evolved later on. Eukaryotes would involve plant and animal cells. And when we look at bacteria, they have cell membranes, they have a cell wall, they have cytoplasm, and they have DNA. They also have ribosomes. And this helps to show us that ribosomes show evolution, right? They've been around since the beginning, that they show common ancestry for all known life. So then, can you compare and contrast bacteria and plant and animal cells or eukaryotic cells? So prokaryotic cells, the name is pro before karyon, before the center of the cell. So before the center of the cell, before the nucleus. 9 out of 10 cells in our body are bacteria. Right? DNA is organized in a nucleoid region, but not encapsulated in its own membrane. And it's one big circular chromosome. Whereas in eukaryotes, DNA is in linear chromosomes. For instance, in humans, we have 23 pairs of them. There are small plasmids or small circular pieces of extra DNA, and that'll be fun in Unit 6. We get to do some experiments with those to try and give bacteria some different properties. They have ribosomes. They have a cell wall, which is made up of modified carbohydrates. They can have tails and capsules and hooks. And then there's different types. And so you could research these as well if you'd like to learn more about bacteria. All right, once again, here are those highlights, those vocab ones. When we look at the unit guide for topic 2.2, like I said earlier, there's a lot of the same ones, ER, mitochondria, lysosomes, vacuoles. The first one was saying, what are they? And these now say, how do they contribute to the function of the cell? And so I will do them both together, right? For instance, the mitochondria, right here, it's saying the function is it's going to help to provide for metabolic reactions or making ATP. Its structure is it has a double membrane, and it's going to have ATP synthase enzymes, etc. All right, and so then there's more here that we'll go over as well. Okay, here is a picture of the cell that I got from Khan Academy. This is a general model, and it's very helpful to have models in biology, but not every cell looks like this, right? Eye cells look very different. They have rods and cones, and so does intestinal cells, etc. But Even though they may look different, they have a lot of the same parts on the inside. So looking at this general model can help us. When we're doing this, when we go through them, I want you to think about this in terms of jobs. And I got this from the great late Miss Foglia about protein production, energy conversion, cellular division, storage and recycling materials, and support and movement. Right? All these are going to contribute to at least one, if not more, of these jobs that a cell must do to be alive. So let's do it. Let's jump in and learn about the different parts of the cell. So here we're going to do ribosomes first. All right. Ribosomes are these structures that are made of ribosomal RNA and proteins. They latch onto messenger RNA, which has been secreted from the nucleus, and they use that to translate messenger RNA into a protein. That protein is going to be a series of amino acids that are joined together inside the ribosome, right? They're joined together in those peptide bonds that we learned about last unit to make a polypeptide or a protein chain. They're brought there by tRNA. Ribosomes that are free floating within the cytoplasm of the cell produce proteins that are used inside the cell or intracellularly. Ribosomes that are studded along the endoplasmic reticulum, like these ones here, will often produce proteins that are exported from the cell. All right, so we did structure and function, right? The function is to make proteins, structure is made of rRNA and proteins. ER. The ER is going to be connected to the nucleus. It's a pathway for protein synthesis. It has its own membrane. It is a membrane-bound organelle made of phospholipids. The rough ER is going to be studded with ribosomes, whereas the smooth ER is not. The rough ER is engaged in protein synthesis, right? Because it has these ribosomes on it. And it's going to, the proteins will be made on the inside here, and then they'll be shipped to the Golgi apparatus to be further modified. They'll be shipped from the ER to the Golgi apparatus in a vesicle that holds them. The smooth ER is going to help make phospholipids, although so does the rough ER, and it helps to detoxify poisons. So cells that deal with poisons will have much larger smooth ERs, and alcoholics have much larger smooth endoplasmic reticulums. Okay, and here is pictured the smooth ER. All right, the Golgi complex is also membrane bound. And here is the Golgi complex. And it's a series, they look like little pita pockets, right? It's a series of membrane-bound organelles that will receive vesicles of proteins from the ER and then modify them. So inside of this compartment that's been compartmentalized, the protein can have carbohydrate markers added to it. It can be refined. It can be changed before it exits the cell. This gives us much diversity in protein structure and function that can be exported. So it's another membrane-bound organelle, and its function is to modify proteins and then prepare them to be shipped out of the cell. They will leave the Golgi apparatus inside of vesicles or little carrying packages that are cellular membranes, merge with the cell membrane here, and then go off into the bloodstream to go do their work. So that is the Golgi complex. We've done the ER, we've done the ribosomes. Let's do the mitochondria. Here are the mitochondria. And the mitochondria, notice how they're about the size of a bacterium. And they're going to be the site of ATP production for the cell. And ATP is going to be the cell's energy. So these mitochondria will take glucose. Well, glucose will be split in the cytoplasm into what's called pyruvate. It will take that pyruvate and it will then turn that into ATP, which is a usable form of energy for the cell. You could think of glucose like a big container of gasoline for your car, right? If you just lit a match to it, it'd be a huge fire. It'd be deadly. You have to step that down. You have to get a smaller unit of energy called ATP. Lysosomes and the structure of the mitochondria, by the way, contributes to its function, right? It's going to have a double membrane. And we're going to go over that in more detail on how it works with the Krebs cycle and the electron transport chain. But it's really going to set up for maximum ATP production. The lysosome are small vesicles that contain enzymes that help to digest dead organelles or broken organelles, a lot of hydrolytic enzymes that can break down things. They're like a digestion packet. So if the cell brings in a large piece of food, it can use the lysosome to help break it down. The lysosome can also recycle older and dead things. So its structure of being a self-enclosed membrane allows it to have more acidic pH, and it allows it to have the enzymes inside of it to break down other things. If the lysosome was opened, then those enzymes that are going to digest old organelles will be all over the place. All right. Chloroplasts will be on the next slide. So, sorry, we'll come back to this one. Well, I had to add in more depth. Well, here it is. Chloroplasts are here. And so they're going to be the sites of photosynthesis inside of the cell. And we'll get into that more later on. Vacuoles, I believe, are also on that page. And these are large storage areas that store water and other enzymes inside of plant cells. So let's go back. One thing that's important in biology is the endomembrane system. And endo means within, and membrane meaning a phospholipid bilayer. And so these are connected and they're all membrane bound organelles. So we're going to start out with the endomembrane system for producing proteins, right? And so here, the nucleus has your DNA. The DNA is stored here. It cannot leave, right? It's too big. It can't fit through these nuclear pores. So DNA is transcribed an enzyme called RNA polymerase to make mRNA, which goes to these ribosomes on the ER, the rough ER. The rough ER then sends the proteins that are made here to the Golgi apparatus via vesicles, so these little carrying molecules. Think about it like little taxis. They carry into the Golgi apparatus, and then the Golgi apparatus will modify them and send them out of the cell. All right. Lysosomes are digestive enzymes like we went over earlier, and here's a picture of them helping to digest food. The lysosomes are part of the endomembrane system. All right, and as well as the vacuoles as part as well, because they store enzymes. Okay, here we go, endomembrane bound. So you might be using Khan Academy or learnbiology.com, or you'll get to use AP Classroom once we're back together as some supplements. And so here it's going to go a bit further in depth on the mitochondria and chloroplast. So let's check them out. And I'm looking at my guide here that says we need to know a fair amount of the items inside the chloroplast. So let's dive in. The chloroplast has two membranes. It has an inner membrane and an outer membrane. It's going to have these thylakoids here that is the site of the light-dependent reactions of photosynthesis. A stack of them is called a granum, that's B1, and in multiple granums would be grana. So granums is not correct. Grana is multiple granum. The stroma or fluid around these thylakoids is where the Calvin cycle takes place, where the products of the light-dependent reactions, which would be NADPH and ATP, are used to help fix carbon dioxide into an enzyme called Rubisco that's around, and it's then going to be made into sugars, right? So the sugar part happens out here. More to come in Unit 3. Let me pause for a second here. So I just checked my guide, which I then just had come up right here. So the one thing I missed was that these thylakoid membranes have chlorophyll inside of them. And the chlorophyll is what's going to get excited by the sun's rays, the sun's photons, and then begin the process of photosynthesis or the light-dependent reactions. All right, the mitochondria. So let's look and see what the guide said. We need to know that the Krebs cycle reactions occur in the matrix, and that this electron transport ATP synthesis occur on the inner mitochondrial membrane. So like I said, many teachers will go over this more in Unit 3, which deals with photosynthesis and cellular respiration. So here's the outer membrane, and here's the inner membrane. And here's a good example of how the structure leads to the function of a molecule or of an organelle. The pyruvate, which is broken down sugar, will get brought in. She's purple. And so it comes in here into the matrix. In the matrix, As many high energy electrons are stripped from it as possible, and it's going to then the carbon and oxygen will be expelled as carbon dioxide that will leave, so CO2 will leave, and they will carry these high energy electrons to this membrane, the inner membrane. The inner membrane has a series of proton pumps on it that are able to pump lots of protons to this intermembrane space. These protons came from the oxidation of glucose. The protons there in the inner membrane space will be able to diffuse through an enzyme called ATP synthase to make lots of ATP. And there, those protons will recombine with oxygen to form water. So, a good example of how the structure of an organelle leads to this function. And let's move on. All right, time for you to quiz yourself for this part of the video so far. Can you compare and contrast prokaryotic and eukaryotic cells? Can you describe processes involving energy conversion, protein synthesis, and the organelles there, and the structure and functions of these organelles? All right, pause the video and have fun. All right, last bit here, we're going to go over why are cells small. And this is a good chance to practice your math skills here for surface area to volume ratio. All right, so for this part of the course, you're going to explain how surface area to volume ratio restricts cell size. And you need to be comfortable using these formulas. They're given to you on the test. You do not need to memorize them, but you do need to be comfortable using them. Okay? You can notice that volume is going to be cubed, whereas surface area is going to be squared. That will help you out in a second. All right, let's talk about this via a lab. And so many teachers do it where they have some type of agar and they have you cut different cell sizes, and the agar has an indicator in it. And you can either put the agar inside of a base or inside of an acid and then watch the time of diffusion. And what we're trying to mimic here is getting oxygen into a cell and carbon dioxide and other wastes out of a cell, right? The cell is the smallest unit of life, so it has to be able to get oxygen to the parts that need it, like the mitochondria. If it cannot do that, it cannot be alive. So using your formula sheet, see if you can calculate the surface area and volume of this cube, the 1 by 1 by 1 cube, the 2 by 2 by 2 cube, and then this rectangle that's 1 by 1 by 8. See what you can do using your formula sheet. Here's what you should have gotten. The surface area to volume ratio for the first cube is 6:1, right? So it'd be like 6 nanometers squared to 1 nanometer squared. So that is the surface area to volume ratio. For the next one, you should have gotten 3. So when we make the cube bigger, notice how the surface area to volume ratio went down. That's because surface area is to the second power, whereas volume is to the third power. So eventually, volume will always outpace surface area, regardless of the shape. And then here, we got 34:8, which is going to be somewhere in the middle. So these are some pictures of the lab, and you can see here for this one, I stained them with bromethanol blue, and then I put them inside of an acid, and slowly it diffused in, and you can measure the rate of diffusion to mimic why cells are synthase small. So here's our chart again. So that for the first one, surface area is 6, volume 1. Surface area to volume ratio is 6:1. The next one, 24:8. And so if you reduce that, that would be 3:1 would be the ratio. 6:1 is the ratio of surface area to volume. Sometimes you do ratios as fractions. So it'd be 6/1. This would be 3/1. And this one was in between. It's 34:8. So the ratio is 4.25:1 is what I got. So if you were to think about this, ask yourself which cells should have the quickest rate of diffusion to get oxygen all the way to the inside, to the very inner part. And hopefully you got the idea that the higher the surface area to volume ratio, the quicker the rate of diffusion. If you have access to it, there's a thing called Explore Learning Gizmos, where there's a really cool simulation on chlorine gas poisoning that happened because of a train wreck, a true story. And it goes through what affects the rate of diffusion and what really causes a concentration gradient. So here I got 29 minutes for this one, 53 and 46. So this is my times, and hopefully it works, right? The 6-to-1 is the quickest rate of diffusion for this cell, whereas the 3-to-1, which was the smallest surface area to volume ratio, took the longest. This is why cells can't be huge. We can't just be one cell. This cell, it would take it too long to get the oxygen all the way to the inside and to get the CO2 or its waste out. So cells are inherently small to maximize surface area to volume ratio and maximize diffusion rates. All right, surface area to volume. Here's another example of it. Notice how chopping these into lots of little cubes really increases the surface area to volume ratio. Same volume, but by making it lots of little cubes, it has a higher surface area. There are many examples of adaptations to maximize surface area to volume ratio. So here's just a picture of the lab. Here's a picture of some adaptations. This is your intestinal lumen cells. And so in here would be like the food you ate. And so here it has all these villi in order to increase the surface area for the rate of absorption of nutrients. Same thing here. This is a root tip cell. And so these are all little root hairs going off of it. And they have little projections going off of it to increase their surface area. So it looks like this. That's for a plant. All right, quiz yourself. Did you get for cell size, why are cells small? Can you perform calculations of the AP Biology formula sheet on surface area to volume ratio? There's some good practice problems for that in AP Classroom. Can you describe adaptation cells have to increase surface area to volume ratio? And that is the end of this video. Take care.